ATP is a molecule our bodies use as energy currency. Most ATP in our body is made using the mitochondrial protein ATP synthase. ATP synthase has two regions, a mechanical F0 region and a catalytic F1 region.
The F1 region features the alpha-beta complex and a hexameric ring structure responsible for binding ATP and inorganic phosphate, catalyzing the creation of ATP and releasing ATP into the mitochondrial matrix. Connected to the hexameric ring is the central stalk, made up of gamma and epsilon polypeptide chains. As the stalk rotates, it stimulates the synthesis and release of ATP molecules. The hydrophobic F0 region of ATP synthase is a proton pore embedded within the inner mitochondrial membrane. It has two main subunits, A and C, that interact together to form the proton channel that allows for the proton movement. The C subunit is in a ring formation and acts as a proton channel that allows hydrogen ions to flow from the inner membrane space into the matrix down their electrochemical gradient. The A subunit has the ion half channels that the hydrogen ions use to bind to the C subunits. It lies on the outer part of the C ring and helps to move those protons across the membrane. The B subunit connects to the catalytic region of ATPase via the delta subunit, which helps to hold the hexamer ring in place and keep it from rotating. The beta subunits are directly responsible for this reaction enzymatically, bringing ATP and inorganic phosphate in contact due to conformational change. This change is a result of rotation of the gamma subunit that connects directly to the C ring via the epsilon subunit.
The ETC is comprised of several proteins that are embedded in the mitochondrial membrane. Electrons flow through these structures, transferring from complex to complex. The flow of electrons helps in the proton movement across the membrane, which ultimately results in ATP synthase making ATP.
The ATP synthase may be regulated depending on how much energy our bodies need. Since oxygen is needed to produce ATP, this would mean that when the body tissue suffers from a lack of oxygen, the ATP synthase will be downregulated, which means the mitochondria will tell the ATP synthase to stop doing its job or do less of it. Upregulation, on the other hand, is increasing the activity of the ATP synthase, and that happens when we exercise. Since more energy is needed for muscle contraction, the mitochondria demands more ATP, so the ATP synthase activity increases.
Some mutations can lead to ATP synthase deficiency, wherein the mitochondrion cannot utilize the proton gradient for ATP synthesis. This can increase the production of reactive oxygen species, which is directly linked to cellular damage, aging, and neurodegenerative diseases. Certain mutations can even result in a condition known as Barth syndrome. Patients with Barth syndrome tend to have an enlarged and weakened heart, muscle weakness, and chronic infections.